Hey guys, how are you? It was great seeing you. This is Miss Muntaha speaking, and this is week 7 of online learning. And today we're going to start with a new activity from nutrients to new molecules. Part 1, the production of new molecules. So let's start. As we studied in previous activities, after we eat food, the food is digested into nutrients. So glucose is the product of starch digestion. Amino acids are the products of protein digestion. Fatty acids and glycerol are the products of fat or lipid digestion. Once these nutrients reach the small intestine, they are absorbed through the blood capillaries that are found in the intestinal wall, in the microvilli and the villi of the small intestine's intestinal wall. So these nutrients will pass through the blood circulation and they will reach a body cell. Any body cell in our body, liver cells, muscle cells, brain cells, skin cells, all the body cells have to receive these nutrients. Now, in the presence of energy, which is liberated by the digestion of these food products that will give us these nutrients, we will have the assimilation of new products. The word assimilation meaning we are going to produce or synthesize or create new products. Now, these products either will be stored in our muscle cells or in our body parts, or we will use these products to make enzymes, or we are going to use them to produce hormones and other products, or they are used as lipids and proteins of the cell structures. So we are going to use these nutrients in order to produce new molecules that have different functions in the body. Assimilation of amino acids, meaning the formation and the joining of amino acids to produce new proteins in our body. So an adult mouse is fed amino acids labeled by a harmless radioactive carbon atom for several consecutive days. The word labeled means that we can track it inside our body because it releases a signal. So this mouse was fed amino acids, which are the product of protein digestion. Later on, when they are examining the new products produced by the mouse, they find a new protein chain, new amino acid chain, and these labeled amino acids are found in the new cells of the mouse. And as you can see, this labeled amino acid is found in the new protein that is found here. This pink is similar to the one that was fed to the mouse. Now, we take a sample of the liver cells from the tested mouse that we talked about in the previous slide, and we measure the radioactivity of the proteins they contain. So as you can see here, the percentage of radioactivity increases from 0 to 100% within 5 days. We conclude that the cells in the body use the amino acids from digested proteins to produce new proteins. Now, the synthesis of new proteins. The synthesis or assimilation or creation of proteins takes place according to the genetic program that's found in each nucleus in a body cell. As you remember, every person has his own DNA, and the DNA is found in the nucleus of every cell. So every person is going to sequence these amino acids to produce different proteins. So if I sequence these amino acids in this way, I'm going to have a synthesized protein 1. Or if I arrange them in a different way, I'm going to have synthesized protein 2. But the arrangement of amino acids is different from an individual to another. So the genetic program, which is unique to every individual and it differs from one person to another, determines the number and the nature of amino acids in each protein and their order in a chain. So no human being has the same protein synthesized because we have different DNA. Now, if you have any question, I would be very glad to answer you. See you soon. I love you all. Please stay safe.